For years, scientists have warned about the rising presence of microplastics in the environment. Tiny plastic fragments found in oceans, food, and even the air we breathe. But now attention is turning to something even smaller and more concerning: nanoplastics. Plastic particles less than 1 micrometer in size. These particles are so tiny that they can enter human tissues, cross cell membranes, and potentially interfere with biological processes. Until recently, detecting them in human body fluids was nearly impossible. But now, a novel method combining optofluidic force and Raman spectroscopy is making it possible to detect nanoplastics directly in blood, urine, and other biological samples, opening a new chapter in environmental health science. So, what are nanoplastics? And why are they so hard to detect? Nanoplastics are the result of plastic materials breaking down into ultrafine particles through mechanical wear, UV radiation, or chemical degradation. Once small enough, they behave less like solid materials and more like colloidal or molecular contaminants. Because they are smaller than the wavelength of visible light, they are extremely difficult to detect using traditional microscopes or filtration methods. Moreover, in biological fluids like blood or saliva, nanoplastics are surrounded by a complex mix of proteins, fats, and cells, making identification even harder. How does the new detection method work? The breakthrough involves a combination of two advanced techniques: optofluidic force manipulation and Raman spectroscopy. Optofluidic force involves using a highly focused laser beam, sometimes called optical tweezers, inside a microfluidic channel to trap and isolate tiny particles in motion. This allows researchers to physically separate nanoplastics from the surrounding fluid in real time. Once isolated, Raman spectroscopy is used to shine a laser on the trapped particle and analyze how the light scatters. Every material has a unique spectral fingerprint, and this fingerprint allows scientists to identify not just that the particle is plastic, but also what type: PULT, PET, polyethylene. You might ask, has this ever been done in living systems before? Until recently, studies of nanoplastics in the human body were limited to indirect measurements or studies on animals. This new approach allows for direct, label-free detection of individual nanoplastic particles in real human samples. That means researchers can now begin tracking how much plastic is actually entering our bodies through food, water, or air, and where it ends up. Why does this matter? Because the health implications of nanoplastics are still unknown, preliminary studies suggest that nanoplastics may trigger inflammation, oxidative stress, or disrupt cell membranes and mitochondrial function. They may act as carriers for toxic substances, or interfere with hormone signaling. But without tools to detect them in the body, it was difficult to study exposure and impact. Now, with this method, scientists can monitor nanoplastic levels in people with different diets, lifestyles, or medical conditions and begin to answer questions that were previously out of reach. Could this technology be used outside the lab? Yes. This platform could eventually be miniaturized into diagnostic tools for clinical use, especially in environmental medicine. It could also be used to screen food, bottled water, and pharmaceutical products for nanoplastic contamination, helping to enforce safety standards and guide regulations. The method uses a tightly focused laser beam within a microfluidic channel to generate optical gradient forces that trap nanoscale plastic particles in suspension. These forces arise from the interaction of the laser's electromagnetic field with the particle's dielectric properties, drawing the particle toward the beam's focus. By tuning the laser power and flow rate, one can selectively trap particles in the 5500 M range, despite background biological debris. The microfluidic confinement ensures high-throughput sampling of body fluids with minimal reagent consumption. This approach concentrates nanoplastics into a sub-micron interrogation volume for subsequent spectroscopic analysis. In spiked serum samples, The system trapped 200 pm polystyrene beads at flow velocities up to 5 pm/s, with over 90% trapping efficiency. It's like using a laser vacuum inside a tiny fluid channel to pick out individual dust specks from a stream of water. Once trapped, the particle is illuminated with a second laser tuned to excite molecular vibrations characteristic of plastic polymers. Inelastic scattering of this light, known as the Raman effect, produces spectral peaks unique to different polymer types such as polyethylene, polystyrene, or polypropylene. The method's high spectral resolution allows discrimination among plastics even when particles are sub-100 nm in size. Background signals from proteins and lipids are suppressed by spectral filtering and time-gated detection. This chemical fingerprinting confirms the identity of each trapped nanoparticle in real time. Trapped nanoparticles from urine samples produced a distinct peak at 1/605 cm1. Matching the polystyrene reference spectrum. It's like shining a colored flashlight on a gemstone to reveal its unique spectral glow and confirm its mineral identity. Microfluidic channels fabricated in glass, or PDMS, enable continuous flow of microliter volumes of blood, urine, or saliva through the optical trapping region. Channel designs incorporate cascaded traps to sequentially sample multiple points in the fluid stream, boosting throughput to thousands of particles per hour. Flow rates and laser positions are computer-controlled to balance trapping stability. With this integration allows automated, hands-free operation compatible with clinical workflows. The small channel dimensions also reduce sample-to-sample cross-contamination. A prototype device processed 50 UL of plasma in under 10 minutes, detecting nanoplastics at concentrations as low as 10 particles/ml. It's like a high-speed assembly line sorting and inspecting tiny beads one by one under a microscope. By combining force-based concentration with sensitive Raman readout, the platform achieves limits of detection down to single digits of nanoplastic particles per milliliter. Signal-to-noise ratios above 20 dB are maintained even for particles below 100 dm diameter. The optical trap enhances local particle concentration by factors of 103-104, effectively amplifying weak Raman signals. Rejection of autofluorescence and thermal background further improves sensitivity. This level of detection surpasses conventional centrifugation plus spectroscopy methods by 2 to 3 orders of magnitude. In simulated saliva, the method reliably detected 50 mm polyethylene particles at 5 particles/ml with over 95% confidence. It's like using a magnifying glass in a dark room to spot a single speck of dust glowing faintly. Spectral libraries of common plastics enable automated classification algorithms to assign each Raman spectrum to a polymer type. Multivariate statistical tools, such as principal component analyses, discriminate subtle spectral differences among similar polymers. Real-time software flags ambiguous spectra for secondary validation, ensuring high classification accuracy. Cross-sensitivity with known standards confirms over 98% specificity across 5 polymer classes. This specificity is critical for assessing exposure sources and health risk correlations. In mixed samples containing polystyrene and polyethylene terephthalate, the system correctly identified 99% of each particle type. It's like a barcode scanner that instantly reads and catalogs grocery items passing on a conveyor belt. Minimal sample preparation is required. Body fluids are diluted with buffer and introduced directly into the microfluidic inlet. No chemical dyes or reagents are needed, preserving the native state of biological matrices. The optical wavelengths used do not induce photo damage to proteins or cells outside the trap focus. Integrated on-chip filters remove cells and large debris upstream of the trapping zone. This streamlined workflow reduces operator time and potential sample loss. A direct injection of diluted whole blood yielded clear trapping and Raman spectra without prior centrifugation. It's like pouring juice directly into a juicer without needing to peel or pre-process the fruit. Quantifying nanoplastic levels in urine, blood, and sputum provides direct metrics of human exposure and bioaccumulation. Correlating these metrics with clinical parameters, such as inflammatory biomarkers, illuminates potential health impacts of nanoplastic ingestion or inhalation. Longitudinal monitoring can track changes in exposure over time or in response to dietary interventions. Population studies enabled by this method will inform regulatory guidelines and public health policies. Early detection of elevated nanoplastic levels could prompt preventive actions to mitigate risks. A pilot study found that industrial workers had 10-fold higher nanoplastic counts in urine compared to office workers. It's like measuring blood lead levels to assess exposure and prevent long-term toxicity. Traditional techniques, such as filtration, electron microscopy, and bulk spectroscopy, require extensive sample preparation and cannot distinguish particles below 500 nm without labeling. The Optofluidic Raman approach provides label-free, in situ detection with minimal handling. Real-time analysis eliminates lengthy downstream processing and reduces potential contamination. The integrated platform also quantifies both size and chemical composition simultaneously. This multifaceted capability accelerates research into nanoplastic distribution in human tissues. A side-by-side comparison showed the new method detected 90% of 100 nm beads, whereas filtration plus FTIR detected only 10%. It's like switching from a manual metal detector to a smart sensor that instantly identifies metal type and size as you sweep it over sand. Ongoing work aims to miniaturize the laser and detection optics into a handheld cartridge-based analyzer for point-of-care use. Integration with cloud-based data analytics will enable remote monitoring and large-scale epidemiological studies. Efforts to expand polymer libraries will cover emerging biodegradable plastics and environmental contaminants. Combining this platform with cell sorting modules could assess intracellular nanoplastic uptake. These advancements will pave the way for routine clinical screening and personalized exposure assessments. A proof-of-concept portable unit successfully performed on-site urine testing at a community health clinic. It's like evolving from a room-sized MRI scanner to a portable ultrasound probe you can carry to the bedside.